My name is Roger Cohn, and I'm Vice President and Director of the Biosciences Initiative. I'm really thrilled to be able to welcome you all here this afternoon to our first community kickoff, celebrating the progress of the first year and introducing our inaugural scientific initiatives. During this first year, we've provided funding already for 6 different programs designed to support the biosciences here at Michigan. We've provided funding to the Michigan Life Sciences Fellows Program, a super postdoc program designed to bring the best and brightest postdocs here. We've helped extend the Biological Sciences Scholars Program, a 15-year-old program that has had a lot of success in bringing top quality assistant professors to the medical school. We've extended that to all the bioscience units and provided funding for that program to give everybody a competitive edge for recruiting assistant professors. We've just recently provided funding to the Precision Health Initiative to improve the diversity and the breadth of the Michigan Genetics Initiative DNA Core resource. In collaboration with the School of Medicine, we have funded the creation of a single website that will contain all of the core labs available to bioscientists. And this is going to launch in the coming weeks. And there's a poster outside on that, as well as the other programs I've just described. And also will be available afterwards to take any questions you have about these programs. We've funded a number of exploratory proposals in the first round of competitions. These are small programs designed to explore new areas of research for Michigan. And again, there's posters on these exploratory programs as well outside that you can look at during the gathering afterwards. But in this presentation here, I'd like you to hear from the directors of the 5 large scientific initiatives that the Biosciences funded this October. These programs truly represent the incredible breadth of bioscience research going on across the more than 11 schools, colleges, and institutes here at Michigan with an emphasis on the life sciences. The funding of these programs will bring 14 new faculty to the University of Michigan, create at least 4 new research cores, and move forward biosciences across the whole university. So I'd like to, at this time, take a moment to recognize and thank all the people who've put a lot of effort in to make this work happen over the last year. I want to thank President Mark Schlissel and Provost Filbert for their continuing commitment to this program and all of their help and assistance. The Biosciences Initiative Coordinating Committee has worked tirelessly to create these RFAs, review grants, interact with faculty across the university. It's been a fabulous committee to work with, and many of the members are here today. Hopefully you'll have a chance to talk with them afterwards. Vice President of Research Jack Hu, he's here today. We've worked closely with his office and members of his office in this entire initiative. Amy Dittmar and her office have provided budgetary assistance to help us manage the budget for this initiative. And the Biosciences Initiative staff, who many of you have interacted with, Jennifer Oren, Donna Ray, and Annie Cadelli. With the support of these people and many more, we've had a strong first year. We're looking ahead to the next year. In January, we'll be announcing some new funding activities, as well as the reissue of the RFA for the large scientific initiatives and exploratory programs. One of the programs that we'll be releasing in January will be a rolling RFA to begin providing resources to the core laboratories, either to improvement of existing core laboratories or creation of new core laboratories. And so you'll probably get emails about these if you're on our distribution list, and of course they'll be announced on our website, and you can sign up for our quarterly newsletter as well. A rough timeline for these RFAs will be announcement in January, letters of intent due April 3rd. May 31st, the Biosciences Committee will let people know which applications are invited back for a full proposal, Full applications will be due August 30th, and we're planning to make the announcements next year by December 1st. So please keep an eye out for future information, and also feel free to contact me or any of the other members of the Biosciences Initiative or Biosciences Committee if you have questions about any of these activities. So I'd now like to welcome President Mark Schlissel to the podium to talk about his vision for the biosciences and to launch today's event.
So good afternoon, everybody. I'll be very brief and a little bit more selfish than usual. This is one of the most exciting days for me since I've been here. And the reason why, when I arrived here nearly 5 years ago, I saw a vast biomedical research enterprise spread across multiple schools. And I saw a breadth of talent in other areas focusing on biological problems as well, in engineering and physics and math and statistics, and even in the Institute for Social Research, people were collecting DNA samples. And the question I asked is, how can, at the campus level, not one school at a time, we stimulate new levels of excellence and importance out of an already fantastic biological sciences community? And it became clear to me that the challenge is, how do we put people together from all these many fields where we've got such outstanding academic excellence to come together around problems? One of the ironies is I end up knowing more people at the institution than folks that have spent their lives and their whole careers here. Because as faculty, most of us work inside of a niche, and at my level, I get to see the breadth of the whole campus. So the idea was to set aside the amount of money that my predecessors might have used to build a new building, a new institute like LSI, for example. And we're blessed with having great facilities, I think. So rather than invest it in bricks and mortar, the idea was to invest it in people, in new projects, and to set up a source of resources that would stimulate people looking to colleagues in disparate fields to come together and do things that really we might only be able to do here. And the hope was not to come up with 5 things that are identical to things that are going on at other universities, but really go after novel, exciting ideas that can tap into the breadth of the institution. So I want to thank Roger and the Biosciences Initiative Coordinating Committee for thinking about how to do this, how to make this real, for investigating what's done in other institutions, and for developing a novel approach to trying to tap into this outstanding breadth of the university. I want to thank all the faculty that did the legwork in developing, identifying colleagues and developing these ideas. And of course, congratulations to the first round of awardees. It's quite cool that Roger and the committee have come up with a breadth of ways to stimulate life science across the campus. And a common feature of all these things is they're not specific to a single department or a single school. They're really designed to bring people together and tap into the synergies that really only a place like Michigan can develop. And I'm really quite confident that after several rounds of this, and we blow through $150 million, we will really have elevated the excitement level and the type of science we do and its impact broadly across the whole institution. And I really look forward to seeing it evolve. So thanks, Roger, and I look forward to the talks.
Well, thank you for those remarks. I'd now like to introduce Steve Sram, our MC for the Scientific Initiative grantee presentations. Steve is Executive Director and General Manager of Michigan Public Media. He oversees Michigan Radio, the NPR News and Information public radio station here at the University of Michigan. Michigan Radio is one of the state's most listened to public radio stations, serving over 500,000 listeners. And under Steve's leadership, the station's local journalism news team has won multiple Edward R. Murrow Journalism Honors, a National Scripps Award, and the prestigious DuPont Columbia National Journalism Award during his 12-year tenure. So take it away, Steve.
Thank you, Roger, and good afternoon, everyone. It's my pleasure to be with you today to introduce the talented members of our Academy who are leading research in a variety of critical and groundbreaking methods. There is much to learn and discover today from their collective work that we're going to hear about in the next hour. Let's get underway with our first project, the Michigan Concussion Center, led by Principal Investigator Steve Imbroglio. Dr. Broglio is co-director of one of the largest concussion studies in the nation, jointly funded by the NCAA and the Department of Defense. This proposal calls for the creation of a comprehensive U of M concussion research center through the recruitment of an additional epidemiologist, a neuroscientist, and a clinical interventionist. Dr. Broglio is currently the professor of athletic training and the director of neurotrauma research laboratory in the School of Kinesiology. He also holds an appointment in neurology at the medical school.
Thanks, Steve, and thank you to everyone for coming this afternoon and spending some time with us to listen to the projects that we've been fortunate to receive. Concussion is an injury that nearly everyone has some sort of connection with. The Center for Disease Control has identified concussion as an epidemic, with nearly 4 million sport and recreation-related injuries occurring each year. The Department of Defense also suffers from a number of concussions, with nearly 85% of their injuries occurring outside of combat situations, suggesting a mechanism of injury that's virtually identical to sport injury. While discussions about banning certain sports are occurring, moms and dads are struggling with letting their children participate because of a lack of information. The Michigan Concussion Center is aimed at producing cutting-edge research that will help inform not only that discussion, but policy decisions and patient care. Different from other centers around the country or around the globe for that matter, the Michigan Concussion Center will take an A to Z approach to the injury, not focusing on one single pre-morbid or post-morbid factor, but everything from beginning to end. Faculty and staff from 13 different units from across the campus cover many of the research domains that you see above. In particular, impact biomechanics. We have phenomenal faculty from physical medicine and rehab that are investigating the location and the magnitude of impact that it takes to cause a concussion, while our engineering colleagues are looking for new materials to mitigate some of those forces and protect our athletes and soldiers. In addition, if we look at the far right-hand side, some of our sports analytics individuals are looking at the economic cost of concussion to the athletes and their careers and what that may mean. What we're most excited about, however, is the 3 new faculty members that we're going to be able to add because of the Biosciences initiative. First, we're interested in hiring an epidemiologist to help us understand the true incidence and population-level outcomes of concussion and its long-term effects. This remains largely unknown to this day. We're also looking for a clinical trials or interventionist to help develop and implement pharmacological, cognitive, and rehabilitative interventions aimed at improving patient outcomes and reducing overall symptom load, recovery time, and mitigating long-term effects. And lastly, we're interested in hiring a basic neuroscientist that uses cell, human, or animal models to identify biomarkers for injury diagnostics, recovery, or the effectiveness of interventions. We're excited about the collaborations that we have, and we're interested and very excited about new partnerships that we may have, placing any of these 3 individuals into various units across campus: microcellular developmental biology, the School of Kinesiology, Michigan Medicine, and a number of different units, nursing, public health, and on. Research, however, is just the backbone of the Michigan Concussion Center. We would be remiss if we neglected patient care. We want to integrate cutting-edge research into patient care and use our clinicians to inform the research team what things need to be addressed moving forward. We've partnered with Michigan Neurosport, housed in the Department of Neurology, which is a sport concussion-specific clinic, as well as physical medicine and rehab for an adult TBI clinic. The CSMA Emergency Department has also partnered with us, and of course, we've partnered with the Department of Athletics, who's been a huge supporter of our work for a number of years. Lastly, we're very interested in public engagement. This is an injury that's discussed at the dinner table almost nightly, and in addition to our traditional online website and Twitter types of feeds, we've partnered with Michigan News to help disseminate our research and educational materials, as well as working with our state and federal members to help develop policy and push that out. And then lastly, we partnered with the Injury Prevention Center to make sure that we're targeting all the right groups and individuals that they work with. Of course, sustainability is always on our mind, and we've partnered with our development office to make sure that we can make that happen. I would be remiss, however, if I didn't mention our incredible team that helped develop the proposal, the executive committee. I'm just the fortunate one that gets to stand here today, but without their help and their support throughout the entire process, we wouldn't be here. Thank you very much for your time. I look forward to meeting many of you when we're done.
So let's just ask one question if we can right now. Steve, how will your program advance the diagnosis and treatment of concussion?
I think one of the things we're most interested in at this point are developing or identifying new blood biomarkers and this is perhaps one of our basic scientists can help along those lines. So you can imagine a pinprick, something almost like a glucose meter, can be used on a sideline or in a combat situation, put it into a handheld device, get a reading, and make a decision on the site.
It's fantastic. It's a fascinating program. Congratulations and
Thank you
good
very
luck on that. Let's move on now to our next funded project, and it is titled From Cells to Atoms, the Future of Cryo-Electron Microscopy at the University of Michigan. Program Director Melanie Oy is joining us right now to speak about the project. Dr. Oy is the Rowena G. Matthews Collegiate Professor in the Life Sciences. She is a Research Associate Professor of U of M Life Sciences Institute and Associate Professor of Cell and Developmental Biology at the U of M Medical School. Cryo-EM uses electrons to visualize frozen samples and is quickly becoming the go-to technique for structural biologists. U of M was an early adopter in this program with the technology here over 5 years ago. The new funding will enable the university to become an international leader in the field and a premier destination for cryo-EM education. Among other things, the funding will help U of M researchers expand their ability to visualize molecular mechanical machines in the intact cells, developing an approach called Cryo-Electron Tomography. Dr. Oy believes that by combining molecular EM with complementary structural, biochemical, biophysical, and genetic methods, as well as embracing a collaborative multi-team approach, that the aim is to address the molecular basis underlying important biological processes. Her research goal is to generate detailed molecular models that will provide biological insight into function. Dr. Oy, welcome, and please tell us more about your current and forthcoming project.
So in biology, form follows function, and this is a principle that holds true on all scales, the wings of birds all the way down to the atomic structure of the DNA helix. Structure biology is a field where we are devoted to trying to figure out how atomic level details translate into really complex behaviors of cells and organisms. When we generate 3D structures of molecules, what it allows us to do is understand the mechanisms that are important in both health and disease. The more detailed structures that we can determine, the easier it is for us to figure out function. Until recently, there were only 2 techniques that could determine high-resolution atomic structures, and those were X-ray crystallography and NMR spectroscopy. But there has been a number of recent breakthroughs in the technique called cryo-electron microscopy, or cryo-EM, that have allowed this technique to start to also generate structures, really high-resolution structures of very dynamic complexes. These breakthroughs have come because we have better microscopes, new cameras, and much more powerful computational approaches to generate these structures. So in cryo-EM, what we do is we purify complexes or take intact cells and put them on a copper grid. And then these are quickly plunged into a cryogen so that we freeze them in non-crystalline ice. And this is called vitrification. And in vitrification, what happens is that the samples are frozen so quickly that all the atomic resolution detail is conserved. And then we can put these grids into an electron microscope and take images. And these images have all the information that's needed to actually generate a 3D structure, with one problem. Biological samples really don't like electrons. And so we have very, very noisy images, which means we have to collect thousands of images to collate these very large data sets of millions of particles. And then we take these millions of particles and use computational approaches to amplify the signal, get rid of the noise, and generate really detailed 3D structures. So, as was mentioned, the University of Michigan was actually a very early adopter in investing in the cameras and the microscopes that are required for this technique. However, even with access to this technology, cryo-EM requires a lot of expertise and education to be able to actually use it. And so this has created bottlenecks for investigators who want to start to adopt this technique to actually study problems in their own labs. And so one of the things that we want to do, and what this initiative is going to allow us to do, is two-prong approach. One, we want to really expand the ability of researchers all across campus to start using this technique to address their own questions. Mike Sinafrocco and I, over the last year, have met with over 40 faculty. They've come from 4 different schools, 14 different departments, and they all have really interesting problems. However, the bottlenecks in how to freeze your samples, image them, and actually determine a 3D structure are almost insurmountable. And so what we would like to do is, one, invest in a microscope that will allow users to quickly screen their samples and also learn how to use the microscope. We want to invest in new technology, robotic technology that should make vitrification much easier. And then we also want to increase our ability to quickly determine whether your purification actually works or not. And that will be done using mass spectrometry. The other thing we want to do is expand our staff so that researchers who actually want to determine a 3D structure can do this in a more efficient manner. And Mike and I will be developing a number of new approaches for training users, hands-on classes for students and fellows to learn how to use the microscope and move from their sample to structure. And also, we now can continue to offer an international workshop on how to actually determine a structure once you get all your data. In addition, the bar keeps moving for cryo-EM. So the next really big thing in this technique is actually taking what we've learned to study purified complexes and actually start studying these complexes in their in vivo context, in the context of the cell. And this is also very, very difficult. And at this point, we actually don't have either the expertise or the technology required. And so this initiative is going to allow us to invest in faculty expertise. In collaboration with LSA, engineering, and the medical school, we're going to hire 2 new faculty members, one who's an expert in cryo-electron tomography and one who is an expert in an approach called correlative light and electron microscopy. Both of these expertise are necessary to move us to the next level. We're also going to invest in the technology that will allow us to mill these really intricate windows into cells so that we can start actually doing the reconstructions of how the molecules look within the cell. And so Janet Smith, Mike Sinafrocco, and myself were the 3 co-PIs on this grant. We're really excited to be able to expand our ability to do cryo-EM here at Michigan and expand all of your guys' ability to do cryo-EM at Michigan. And we're also very excited to make sure that we can stay at the cutting edge of this technique.
So I think everybody in the room here knows we love it when Michigan is clearly better than Harvard at anything, and like cryo-EM and football. And so how would you compare our cryo-EM program to theirs, and what makes Michigan a leader in this field?
So actually, Michigan invested in this technology 5 years ago, where Harvard, they just got these microscopes a few months ago.
Okay.
So we were way ahead of Harvard at this approach, which is why I'm here.
That's right, and we will be miles ahead as this funding gets underway. Congratulations. I think I'm going to walk over here. I'm going to let our next speaker, which is Alan Burton, I'll let you have the microphone, and I'm going to introduce you here. Now we're going to learn about our next project award made to the Institute for Global Change Biology. The Institute is led by Principal Investigator Alan Burton, PhD, a professor of ecotoxicology in the School of Environment and Sustainability, and he's a professor in the Department of Earth and Environmental Sciences in the College of LS&A. Global change biology seeks to understand the biosphere's responses to human activities. Human-caused global changes include climate shifts, land use conversion, the release of pollutants, and species introductions. Dr. Burton states that every week we are having dynamic climate change events occur, and we can't just continue to observe it, but we have to be predicting what the effects will be. The new Institute for Global Change Biology will foster research to understand and forecast the interactive effects of global change drivers on organisms and ecosystems. The new funding serves as a planning grant to launch the program. A larger, more comprehensive application will follow. Dr. Burton, thank you for being here today. Congratulations on this new award. Tell us more about the new institute.
Thank you. Well, first I want to acknowledge my excellent co-PIs, Inez Benez, who's with me in Cs, and Allison Steiner from Science and Climate. um, and Climate and Space. It's a very complicated department name. And then Newt Nadelhofer from EEB and the Biostation. Second, I want to thank our president, provost, and VP Kahn for making all of this happen. It's tremendously insightful. Unlike my esteemed colleagues here, I'm heading a project with this team that's may not meet its objectives, frankly. The problem is so huge, and there's so many other players involved in climate change that we have no control over. It's going to be difficult to win, but we're going to make our best stab at it. I'm just a laid-back country boy from Arkansas, and I don't get excited about much. But as of last week, I started getting really excited, or alarmed, I guess is a better word. A month ago, the Intergovernmental Program on Climate Change came out with a special report, they didn't wait, that uses some data that they've come up with more recently that makes their predictive models more accurate. And it's predicting effects, climate-related effects, occurring much faster than we had hoped. Last week, the fourth National Climate Assessment came out, and that's the work of 13 federal agencies in our country. They basically supported the findings of the IPCC, but they also addressed the dire social and economic consequences that were going to hit the U.S. And then this week, a hallmark paper came out of the Global Carbon Budget Project. Over 100 scientists from 50 different multi-sector institutions confirming the previous two reports, but saying something that really is quite alarming, and that those horrible things that we are hearing are going to happen at the turn of the century are going to actually start happening this decade, which is quite disturbing. I could stand here for an hour and talk about the disturbing things that are happening in climate change, many of which you've heard. The greenhouse emissions continuing to increase to record levels. The temperatures, we're breaking the records every year for temperatures, and I thought the temperature records are supposed to last for a while, but they're being broken every year. Ocean acidification, we've lost 20% of our coral reefs, others are dying. We're losing charismatic species, and we're losing non-charismatic species. We've lost 60-fold, not 60%, 60-fold of our insect population since the 70s. The insects are the base of our ecosystem. They're making the ecosystem services that help humans happen, and we're losing them at an unbelievable rate. On top of that, we've, of course, got the human health issues to contend with, which are going to be impacting the developing countries most, those that live around coastal areas. The infectious diseases that are carried by insects are expanding and are going to be expanding to the new areas of the world that are warmer, where they don't have any predators, anything to stop them. Waterborne disease, which was going down, is going to be increasing because of increased flooding, particularly in developed countries. And if it's true to form from the past, 90% of the people who die will be children. Right now, we're losing 1.7 million people a year to waterborne disease, and that's probably going to increase. I'm reminded of a quote from one of the famous and insightful movies, one of my favorites, Ghostbusters. Do you remember the scene when Dr. Peter Venkman told the mayor of New York City that we are headed for a disaster of biblical proportions? He said, biblical? Yes. Old Testament wrath of God kind of stuff. And seriously, I never thought I would witness such things. I'm sad that you're going to witness such things. We don't want our children and grandchildren to experience this. So I've given you some really depressing examples of known knowns. Donald Rumsfeld reminded us to think about the unknown unknowns. Where those have popped up of recent, they've been totally unexpected, and they've been scary, quite frankly, because they tend to be synergistic. In the interactions, the effects are worse than we would have predicted. So what are we going to do about this? How are we going to get past all of this really bad stuff? Believe it or not, there's no university in the world that's doing a good job of this, a cohesive job of looking at these many, many problems. The efforts are highly piecemeal, and that's not the way you attack wicked problems in a piecemeal effort. So as President Schlissel said at the beginning, you have to reach across campus, you have to reach across units to attack these incredibly difficult problems. So I'm really proud of the way our university is going to get at that, and it's going to try to, we're going to try to attack these regional to national to international problems in a predictive mode so that we can understand these known and unknown linkages and hopefully translate that science better to the public and to policymakers. So that we can really make a difference. This next year, we're going to hire one of the top people in the world, I'm sure, to take over my job and lead this institute. That person will then lead the hiring of an additional 4 faculty, and these faculty will have cross appointments across campus. We're going to start soliciting ideas for action groups, which are really mini think tanks, so that we can get people working across campus, and we'll seed those with postdocs, just a lot like the MCube program is, to make all of those things happen. And the science outcomes, I think, are going to be fantastic. Because we've got such incredible people on this campus that just don't happen to be working together as well as they could. So we're going to work across campus synergistically to build global change expertise and make a difference. We're going to be producing the future leaders that are going to be tackling the insidious outcomes that we've been talking about. I'm convinced that because of the kind of people we produce, that they will go a long way toward this wicked problem and make the world a better place for our children and grandchildren. We will be the best. Thank you.
Alan, let's take one question from the work that you're about to do. You know, we're reading every day, and you've just mentioned all the changes in the environment, the coral reef die-offs, the devastating forest fires in California, the massive decline in the insect numbers you just talked about. How will your program prioritize what to focus on?
Well, we're coming at it from a modeling standpoint. So the majority of the people that we're hiring are going to have different expertise in environmental modeling. And with all of the data that's being produced these days, I think we're going to probably try to tackle predictive ways initially in the terrestrial area, because there's not a lot going on there, and then probably swing into the aquatic area, which is my love, and where there's a lot more data. And then after that, we'll leave it to our new superstar to decide.
Very good. It's sombering but exciting. Congratulations. Thank you. Right now, we have our 5 scientists on the stage, and we'll just take a brief interlude here to ask a question that we can circulate among the 5 of you. Thinking about the programs that you're about to undertake, Has this stimulated any conversations among you about collaborations within this cohort? In other words, how your work might be cross-pollinated across the university? I know that you'll be working with a number of other departments, but what about your projects working with each other? Any thoughts on that?
The answer is absolutely. Nils and I just had a meeting this morning to talk about synergies and collaboration. I think you and I spoke during our practice talks, and Melanie, we're already working collaboratively with Cryo-EM facility. This man, you know, we got to figure it out.
Yeah, I'm the odd one out.
So you all agree that there'll be some opportunity for that to commingle and bring some of your science, each one of your specialties, to each other's programs in some capacity or another. Well, that's the exciting part about how this is funded and how this global science initiative will be useful across the university, not only in your individual studies, but with the additional faculties that you bring, faculty that you will bring to the campus, the laboratories that will come online, the core laboratories, and of course, Melanie's very impressive camera that will be coming so that we can do all this electromagnetic study as well. So thank you for your thoughts on that. Let's continue now with our next awarded project. It is titled Expanding Natural Products Drug Discovery at the University of Michigan. The program director and principal investigator is David H. Sherman. He's the research professor at the U of M Life Sciences Institute, and he is the Hans W. Voltaik Professor of Medicinal Chemistry at the U of M College of Pharmacy. He is a professor of microbiology and immunology at the U of M Medical School and a professor of chemistry in the College of Literature, Science, and the Arts. David Sherman has created unique natural products discovery programs based on the collection of about 5,000 microbial strains and the integration of some 40,000 microbial extracts into a high-throughput screening laboratory available to all U of M investigators. The new funding will enable recruitment of 3 new faculty, create a natural products discovery core laboratory to identify bioactive metabolites. This initiative will also provide the synthetic biology and microbial engineering needed to optimize active compounds as discovery tools and the potential for drug development. Once again, more fascinating and essential science being funded by the Biosciences Initiative. David, tell us how this is going to improve our research in this area.
Okay.
Here you are, sir.
Thank you so much. So it's really great to be here, and I'm excited to tell you about our initiative, which includes 6 stakeholders: the College of Pharmacy, LSNA, the Medical School, Engineering, and Dentistry. And as you can see here, we have a beautiful photograph of a marine organism. Over the last 15 years, we've been developing an exciting library of natural products which provides chemical diversity. And we've been slowly building our capabilities, expanding our collaborations. And the initiative that we're envisioning and that we're already embarking on includes 2 major expansions. One is to fill in some gaps by hiring 2 junior faculty, 1 senior faculty member. We're very excited to focus on how natural products and the microbiome impacts human health. That's one very important area. Another is plant medicinal agents. So many plants over thousands of years have been essential for improving human health. We all know of some of the molecules that have come from microbial sources like penicillin and erythromycin and tetracycline. And these molecules continue to provide major sources of medicinal agents and pharmaceuticals. About 50% of what is now FDA-approved pharmaceuticals are either coming from natural products, inspired from natural product molecules, or are derivatives of natural products. And we've only scratched the surface. So these 3 faculty will fill in some of the gaps to take us from a group of faculty and collaborators that are excellent to a spectacular situation that will give us national, in fact, global leadership. One critical aspect of that, which is going to be led by my colleague, Dr. Ashu Tripathi, is the Natural Products Discovery Core. And I show you over here, we're going to be developing not only more high-throughput capabilities, high-throughput genomic sequencing of our microbial library, fingerprinting of our extract library, growing these capabilities, and also integrating new technologies, including artificial intelligence and machine learning, to help us determine the very important structures that make up these new pharmaceutical agents. We're also going to be integrating plant extracts, and we're also going to be continuing to discover and develop new microorganisms and sources of microorganisms to feed our chemical diversity library. And the goal of going straight from genome sequences directly to predicting chemical structures so we can use new algorithms and data science to facilitate our work. So we're super excited to grow our capabilities and our collaborators. You see up here, we have over 30 right now. We're super happy to be able to implement this initiative and grow this number of collaborators in a huge way. So we're going to get started immediately, and we want to be able to talk to as many of you as possible who are interested in drug discovery and natural products, and begin the process of developing new technologies, discovering new pharmaceuticals. Thank you very much to the Bioscience Initiative.
Thank you, David. So I've read earlier that there are over 5,000 microbial species that you have collected from interesting places all over the world, like Costa Rica and Peru and the Himalayas. And the question that I would ask is, aren't microbes everywhere? Aren't there interesting species in Sheboygan or Poughkeepsie or Brighton?
There are, and we're going there. So the biostation. The biostation is a fantastic place to collect new cyanobacteria. We didn't know this until recently. The biostation essentially contacted me and said, hey, you know, we collect blue-green algae up here, and maybe you want to take a look. So I went up and put on my waders. A couple of my students went this past summer, brought back fantastic samples right here in our backyard. We can start collecting too.
Very good. All the exotic places aren't too bad either.
No, we
Very good. Well, thank
especially
you.
in the winter.
That's right.
Yeah.
Congratulations on your program as well. Our last grant award today is for the proposal titled RNA Biomedicine, an engine for synergy, excellence, and global leadership in Michigan. Program Director and PI Nils Walter is a professor of chemistry and biophysics in LS&A and a professor of biological chemistry in the medical school. Recent discoveries in biomedicine have revealed that ribonucleic acid, or RNA, is critical to most aspects of human health, and that its misregulation is responsible for many diseases. This project seeks to rapidly expand U of M's nascent Center for RNA Biomedicine through the hiring of 5 faculty members, the creation of 3 core laboratories, and the funding of pilot and fellowship programs. Dr. Walters has told us that with this funding from the Biosciences Initiative, they'll be able to expand collaboration across all of campus to include colleges and schools involved in the biosciences. The project proposal envisions that these investments will propel U of M to the forefront of RNA biosciences in the nation. To understand these dynamics, the center will combine state-of-the-art biochemical, molecular biological, and biophysical approaches. So, Nils, there is an exciting future to explore surrounding RNA discoveries. Tell us more about how this work will inspire entirely new therapeutic disease intervention approaches. Nils. Thank you.
Thanks, Steve. Is this working, actually? Right.
Here, I'll take that. Thank you.
It has been said that the science of today is the technology of tomorrow. And perhaps nowhere is that more the case than with the RNA discoveries that are currently going on and are being a gateway into precision medicine. My name is Nils Walter, as you've heard from Steve, but I'm only one representative of the 150 members of our Center for RNA Biomedicine, 150 faculty members, that endeavors to utilize and leverage the discoveries in RNA biology that are being made here right on campus and translate them into medical technologies. As you may know, RNA is ribonucleic acid, or the cousin of DNA molecules, and as such, an important molecule of information in cellular life. As such, in fact, the Human Genome Project has discovered that over 90% of the human genome of our DNA blueprint is actually not coding for proteins, as was previously thought, but surprisingly is instead coding for RNA. This has led now to many opportunities of new discoveries in precision medicine in particular. As an example, research in the last few years into RNA splicing, a process by which RNA molecules are put together in new ways, has discovered that an aberrant RNA can be targeted by a drug molecule. This RNA molecule is a key reason for SMA, or spinal muscular atrophy. And it turns out that by targeting SMA with this drug, for the first time, toddlers who were prone to, with SMA, have severe disabilities and death as a consequence, can now walk again for the first time. In another example, a company that was founded only 8 years ago, Moderna Therapeutics, is now valued at $10 billion based on their technology to use RNA itself as a drug in making new vaccines that can be readily and rapidly adapted to new strains of virus that are emerging in record time, faster than ever before. It is these types of discoveries that we want to harness here from the University of Michigan campus and translate into precision medicines. In fact, in 2016, a dedicated group of scientists combined and assembled 7 M-cubes into one block that really had as goal to bring people together across all the schools and units of campus and have them work together to make this a possibility and a real opportunity that we can take advantage of. And so with bioscience funding, we now can hire 5 additional faculty colleagues, as you've heard from Steve, that specifically will be hired with express expertise in helping us and all of us collectively to translate our discoveries in RNA biosciences into real medicines, into drugs that can help people in real life. The goal is also supported by opening up 2 open access facilities that have cutting-edge research tools that were developed right here on campus in nascent RNA sequencing, as well as single molecule biophysics. Together with educational outreach through bi-monthly seminars and an annual symposium, we hope to really engage all of you, hire across all the biosciences units, colleges, and schools of the entire campus, and bring everyone together to really make this a reality, that we can be the powerhouse in RNA bioscience discoveries in the country and in the world. So with that, I want to thank, of course, the Bioscience Initiative and all of you for your attention. Thank you.
All right, Niels, so all along, many have thought that RNA was just a boring molecule, didn't do much, but now it's allowing the information in DNA to be made into proteins. So what you're telling us is that RNA biology is interesting and that RNA molecules can be drugs or possibly drug targets.
Exactly. So after the Human Genome Project, when people had still expected that proteins would be all that the genome is coding for, there was a lot of consternation because people thought, I mean, the rest that is not coding for protein, only 20,000 encoded proteins, the rest, 98%, must be junk DNA. And there's even the term still found on the internet. Turns out that most of that is made into RNA, and we can harness, by learning how these RNAs work, these RNAs directly as drug targets or as medicines themselves. So yes, there's a lot of opportunity and potential for the future.
So a whole new RNA horizon on the front here at the University. Congratulations and welcome again. So you've heard about some very exciting programs that are going to be underway thanks to the Biosciences initiative. And this is just the beginning of a long stretch of excitement and research that is going to be launched because of the resources that are available today through the Biosciences initiative. I want to express my thanks to Roger Cohn, our host and director of the Biosciences Initiative, and to his senior project manager, Jennifer Oren, for the opportunity to join you here today and for this celebration of the first scientific initiatives. Congratulations to all of our program directors and their fascinating work, and thank you for coming.